In my presentation, I did exactly did the experiment. All I had to do was look up information that could be helpful for someone else in their experiment. And the topics I'll be talking about today: epigenetics, multiple sclerosis, distributed epigenetics, multiple sclerosis, data collection, and data analysis using Excel and results. Epigenetics is the study of changing organisms modification of genes, and rather than the alteration of the gene code itself. And basically, sequence is it's like a change in your physical appearance rather than a genetic material, and it's like a natural change and occurrence, but can also be influenced by several factors like age, environment, and the disease state. And histone modification is one part of epigenetics, and basically what it does is when your DNA wraps around histone proteins, and the coils in turn gets tighter, and they become nucleosome to chromatin to chromosome. DNA methylation is when one group, a methyl group, are added to the DNA. So basically, it's getting transferred from one thing to the DNA. And microRNA dysregulation is in the normal cell. In the normal cell, the CJ island is unmethylated and the chromatin associated with active histone modifications. In the cancer cell, CPG island, hypermethylation and repressive histone march lead to epigenetic silencing of the gene, this one. And actually, some microRNA genes are silenced through repressive histone modification rather than the DNA methylation, which is this one. And multiple sclerosis is a long-lasting disease which can affect your brain, spinal cord, and optic nerves in your eyes. And what it does, it causes problems with vision and balance and muscle control and other basic body functions. And what it does is when the immune cell, it attacks a fatty material called myelin that's wrapped around the nerve fiber. And by doing that, it damages the nerve cell from sending signals to like other parts of your body, like the muscle fiber in your body. And that will cause problems with your movement and every other thing. And this is a graph showing how it works. The immune cell, it attacks this right here. And since that happens, the nerve cells signal is blocked or damaged, and they can't get through to the muscle fiber. And so symptoms you may have can be fatigue, numbness, or tingling in parts of your body, bladder, bowel, or bowel control issues. And this is a chart of epigenetics in multiple sclerosis. So basically, this is for the people who want to do research on it more, and they can learn more about it. So another example, DNA methylation is basically the same thing showing microRNA dysregulation. So it basically tests that's run on different samples, and it can help people in further research on multiple sclerosis in the different parts. And so this is the raw data, so it's basically a file we found, and we used it to help them in further research. And what we had to do was clean it. And what my mentor mean by that was we take the information we need out of it and put it in a table that we could use, and this eliminate all the excess data that we don't need. Here's an example. So we take the samples, GDS, and organism that it's been used on, and how many samples there are of each. And these are questions to ask yourself so you can know what to do, and how can it help you. And after you finish this, what you should have in the end when you finish all the researching and gathering information. This is a reference, and I like to acknowledge people for letting me be a part of this program and have opportunity to learn something about I did not know. And Dr. Mandal, Dr. Angela Peters, Dr. Bertie, Jared Epscor, Keanu Ware, and Edward Simpson, mentors, friends and family, and Calhoun University.
And you had to go ferret out all that data. So could you get back to one of the slides that you showed a whole lot of data before you cleaned it? All right, now this is kind of, what does that mean?
This is telling you the information and like the it's like the organism itself is saying like what you gonna actually need in your research, and like all this other access information you don't really need that specifically, but yeah, it can help you.
Correct. So it's on one that he's gonna use, the rest of it just like data for something else, but not for his specific project. Thank you. Any other?
Bioinformatics, and you're the only one that did bioinformatics, and nobody else has a clue what you did?
If you really know it, you got to like, pay attention what the mentor was telling you. You couldn't miss anything, because if you miss, you might miss out on something, and it could like help you, and it would have been like important information you would have needed. And shit, if you ain't pay attention, you would never.
Excellent.
And like if they wanted to find a cure for it, or like make some type of medicine for it, they could use this information to help them in their research.